Let's say you're working in an epidemiology lab. There's been an outbreak of E. coli 157H7 strain of bacteria, and you seem to have localized the bacterium to a commercial kitchen. Your boss tells you that they want you to find where or what surfaces in that kitchen are contaminated with this bacterium. How could you do that? Well, one way that you could do it is you could take sterile cotton swabs into the commercial kitchen, moisten them with sterile media, and then swab a number of different surfaces. But how could you determine whether E. coli O157H7 is on your swab versus, say, Staph aureus or Micrococcus luteus or Lactococcus lacti or any other number of commonly found bacteria in a kitchen environment? In this video, I'm going to talk about how you could separate out that E. coli and possibly detect it. One of the simplest ways of separating bacteria for later study is to do what's called the streak plate technique. In this technique, you apply bacteria to an agar plate and then you gradually and systematically spread them out so that you wind up eventually with individual colonies. This is the most common technique, and it's the 4-quadrant technique. So I'm going to describe this one, but I have these other plates here so you can see that the 4-quadrant technique is not the only technique. There are others. In the 4-quadrant technique, you would take an inoculum. So usually we wouldn't use a sterile cotton swab for this, but we'd use an inoculating loop. So we'd apply bacteria here at the very top of the plate, and then we'd spread that out with an inoculating loop, going back and forth from side to side, making streaks as close as we can to each other, but not going backwards. So we always go in one direction. Okay. Then we would flame our inoculating loop. Then we would drag a few bacteria from this first quadrant down here and spread those out to make a second quadrant. Next, we would flame our loop again, drag some bacteria from the second quadrant, and spread those out to make the third quadrant like this. And then sometimes people flame their loops again, or they simply carry bacteria that are already on their loop, and they use those to make quadrant number 4. And by the time you get to quadrant 4, you should have individual colonies. Each colony represents one bacterium or a short chain of bacteria that grew several thousand times to make a little clump of cells that we see as these distinct circle forms. Now, I wanted you to have the true experience of being in one of my classes. You get to see some bad Foster art here, but hopefully you can get the idea of what I'm trying to explain. So what we would do is we would label a petri dish for this experiment. We would put where the isolate comes from, probably our initials, what type of medium we're streaking onto, growth temperature, the usual things that we've already talked about in a previous lab. We would light our Bunsen burner, and then when we do our streaking, we would always we want to keep our petri dish close to the Bunsen burner. You see, what happens when we light the burner is that it heats the air and hot air rises, right? So as this hot air is rising away from the flame of the Bunsen burner, it's providing almost like a barrier or a sterile field that keeps cold air out here from falling into our petri dish, if that makes sense. So we always want to work near our Bunsen burner under the sterile field. The other thing we would do is, as we're streaking our plate, we want to hold the lid of the plate close to our agar. We only want to open the plate just enough to get our inoculating loop in and do our streak. This is to further protect the agar from contaminating bacteria that might be coming from the environment around us. So one thing I always tell students is that when you're streaking a plate, you are Fall River. You want to stay near the Bunsen burner, which is Dartmouth. And if you're streaking all the way out here in Seekonk, then you're open for contamination. You want to stay away from Seekonk, stay close to Dartmouth. Other things that we want to do while we're streaking plates. We never want to talk over the plate because bacteria that's on our saliva is going to come out of our mouth and land in the agar. You don't want to sneeze or cough around the plate for the same reason. You also don't want to remove the plate from your sterile field while you're streaking. Something else that I'm kind of particular about is I want to keep my fingers away from the openings on the plate. I don't want to leave bacteria there because the bacteria will be that much closer to the agar and contaminating my plate. After we've streaked the plate and we put it in the incubator, we always want to put our plates in the incubator upside down so that any condensation that might form in the plate forms on the lid and not on the agar where our bacteria are. That also can lead to contamination. This is another example of how you could do your streak plate. So again, you start in quadrant 1 with a bit of bacteria on your inoculating loop, and you're going to streak those bacteria back. I usually go back and forth. This diagram has you going in one direction. Flame your inoculating loop, then streak down into a second quadrant. Notice on this diagram they're coming 4 times down from quadrant 1 into quadrant 2, but that's okay. Then flame your loop. Here's quadrant 3. So we're only taking a few bacteria from the previous quadrant into the next quadrant. Flame our loop, and then quadrant 4. And the way I was trained, you always use as much of the plate as you can. So I probably would not stop here. As the diagram says, I would probably take some bacteria and go here into this open area and try to use as much space as I can. The one thing you want to be careful of is that you don't go into a previously streaked quadrant, because then you're essentially bringing bacteria from that quadrant where there should be more bacteria into the quadrant that you're working on where there should be fewer bacteria. So you're doing the opposite of diluting, you're concentrating. So here's an example of a streaked plate. Would you say this successfully separated bacteria so that we get individual colonies? I would say no. If you look here, you can see that all of these colonies are really close together. Even if we used an inoculating needle to remove bacteria from one of these colonies, it's unlikely that we wouldn't actually touch a second colony. So I would say that this is not a successful plate. Why isn't it? Clearly, we have picked up bacteria from the previous quadrants, but we've picked up too much bacteria. So much so that by the time we get here to the end of quadrant 3, we can't really separate the bacteria with the space that we have remaining. So what I would do personally is I wouldn't go back and forth into the previous quadrants quite as many times as this experimenter did. I might go back 3 times, but certainly no more than 3 times. You can see they went back 1, 2, 3, 4, 5, 6, 7, at least 7 times. And then what's even worse is they have crossed quadrants here, so they're not exactly diluting the bacteria very well. So they went back too many times, crossed quadrants, and that led to too much bacteria in the third streak. Now that you've seen this, I've posted 2 videos to your course space. One demonstrates poor technique. The second one demonstrates slightly better technique. Watch these videos, make notes of what's good and what's bad, and then take the review quiz for this lab. It's in your lab folder.